Epithelia, the outer surface of the body and the luminal surfaces of cavities within the body are lined by one or more layers of cells that completely cover them. Such layers of cells are called epithelia, singular equals epithelium. Epithelia also line the ducts and secretory elements of glands, which develop as outgrowths from epithelium-lined surfaces. Classification of epithelia: an epithelium may consist of only one layer of cells when it is called a omlayerector or simple epithelium. Alternatively, it may be mltilayerector stratified. Simple epithelium may be further classified according to the shape of the cells constituting them. 1. In some epithelia the cells are flattened, their height being very little as compared to their width. Such an epithelium is called a squamous epithelium. 2. When the height and width of the cells of the epithelium are more or less equal, i.e., they look like squares in section, it is described as a cuboidal epithelium. 3. When the height of the cells of the epithelium is distinctly greater than their width, it is described as a columnar epithelium. Multilayered epithelia are of two main types. In the most common type the deeper layers are columnar, but in proceeding towards the surface of the epithelium the cells become increasingly flattened, or squamous. Such an epithelium is described as stratified squamous. It may be noted that all cells in this kind of epithelium are not squamous. In the other type of multilayered epithelium all layers are made up of cuboidal, polygonal, or rounded cells. The cells towards the surface of the epithelium are not flattened. This type of epithelium is called transitional epithelium, being transitional between unilayered epithelia and stratified squamous epithelium. As transitional epithelium is confined to the urinary tract it is also called orothelium. A third, rather rare type of multilayered epithelium is made up of two or more layers of cuboidal or columnar cells, stratified cuboidal, or stratified columnar epithelium. Lastly, in some situations a columnar epithelium which is really single-layered may give the appearance of a stratified epithelium. Such an epithelium is referred to as pseudostratified columnar epithelium. The various types of epithelia named above are considered further below. All epithelia rest on a very thin basement membrane. Squamous epithelium. The cytoplasm of cells in this kind of epithelium forms only a thin layer. The nuclei produce bulgings of the cell surface. In surface view the cells have polygonal outlines that interlock with those of adjoining cells. With the junctions between cells are marked by occluding junctions, the junctions are thus tightly sealed and any substance passing through the epithelium has to pass through the cells, and not between them. Squamous epithelium lines the alveoli of the lungs. It lines the free surface of the serous pericardium, of the pleura, and of the peritoneum, here it is called mesothelium. It lines the inside of the heart, where it is called endocardium, and of blood vessels and lymphatics, where it is called endothelium. Squamous epithelium is also found lining some parts of the renal tubules, and in some parts of the internal ear. Columnar epithelium. Seen that in vertical section the cells of this epithelium are rectangular. On surface view, or in transverse section, the cells are polygonal. In keeping with the elongated shape of the cells, the nuclei are also frequently elongated. Columnar epithelium can be further classified according to the nature of the free surfaces of the cells as follows. A. In some situations the cell surface has no particular specialization, this is simple columnar epithelium. A. In some situations the cell surface bears cilia. This is ciliated columnar epithelium. B. In other situations the surface is covered with microvilli. Although the microvilli are visible only with the light microscope the region of the microvilli is seen as a SFR/effect border, when the microvilli are arranged regularly, or as a brush border, when the microvilli are irregularly placed. Some columnar cells have a secretory function. The apical parts of their cytoplasm contain secretory vacuoles. Simple columnar epithelium, without cilia or microvilli, is present over the mucous membrane of the stomach and the large intestine. Columnar epithelium with a striated border is seen most typically in the small intestine, and with a brush border in the gallbladder. Ciliated columnar epithelium lines most of the respiratory tract, the uterus, and the uterine tubes. It is also seen in the efferent ductules of the testis, parts of the amygdala and auditory tube, and in the ependymol lining the central canal of the spinal cord and the ventricles of the brain. In the respiratory tract the cilia move mucus accumulating in the bronchi, and containing trapped dust particles, towards the larynx and pharynx. When excessive this mucus is brought out as sputum during coughing. In the uterine tubes the movements of the cilia help in the passage of ova towards the uterus. Secretory columnar cells are scattered in the mucosa of the stomach and intestines. In the intestines many of them secrete mucus which accumulates in the apical part of the cell making it very light staining. These cells acquire a characteristic shape and are called goblet cells. Some columnar cells secrete enzymes. Cuboidal epithelium. Cuboidal epithelium is similar to columnar epithelium, but for the fact that the height of the cells is about the same as their width. The nuclei are usually rounded. A typical cuboidal epithelium may be seen in the follicles of the thyroid gland, in the ducts of many glands, and on the surface of the ovary, where it is called germinal epithelium. Other sites are the choroid plexuses, the inner surface of the lens, and the pigment cell layer of the retina. An epithelium that is basically cuboidal, or columnar, lines the secretory elements of many glands. In this situation, however, the parts of the cells nearest the lumen are more compressed, against neighboring cells, than at their bases, giving them a triangular shape. A cuboidal epithelium with a prominent brush border is seen in the proximal convoluted tubules of the kidneys. Pseudostratified columnar epithelium. In usual classroom slides the boundaries between epithelial cells are often not clearly seen. In spite of this we can make out what type of epithelium it is. This is because the shape and spacing of the nuclei gives a good idea of where the cell boundaries must lie. Normally, in columnar epithelium the nuclei lie in a row, towards the bases of the cells. Sometimes, however, the nuclei appear to be arranged in two or more layers giving the impression that the epithelium is more than one cell thick. The reason for this will be understood easily from it is seen that there is actually only one layer of cells, but some cells are broader near the base, and others near the apex. The nuclei lie in the broader part of each cell and are, therefore, not in one layer. To distinguish this kind of epithelium from a true stratified epithelium, it is referred to as pseudostratified columnar epithelium. A pseudostratified columnar epithelium is found in some parts of the auditory tube, the ductus deferens, and the male urethra, membranous and penile parts. A ciliated pseudostratified columnar epithelium is seen in the trachea and in large bronchi. Stratified squamous epithelium. This type of epithelium is made up of several layers of cells. The cells of the deepest, or basal, layer rest on the basement membrane. They are usually columnar in shape. Lying over the columnar cells there are polyhedral or cuboidal cells. As we pass towards the surface of the epithelium these cells become progressively more flat, so that the most superficial cells consist of flattened squamous cells. Stratified squamous epithelium can be divided into two types, non-keratinized and keratinized. In situations where the surface of the epithelium remains moist, the most superficial cells are living and nuclei can be seen in them. This kind of epithelium is described as non-keratinized. In contrast, at places where the epithelial surface is dry, as in the skin, the most superficial cells die and lose their nuclei. These cells contain a substance called keratin, which forms a non-living covering over the epithelium. This kind of epithelium constitutes keratinized stratified squamous epithelium. Stratified squamous epithelium, both keratinized and non-keratinized, is found over those surfaces of the body that are subject to friction. As a result of friction the most superficial layers are constantly being removed and are replaced by proliferation of cells from the basal, or germinal, layer. This layer, therefore, shows frequent mitoses. Keratinized stratified squamous epithelium covers the skin of the whole of the body and forms the epidermis. Non-keratinous stratified squamous epithelium is seen lining the mouth, the tongue, the pharynx, the esophagus, the vagina, and the cornea. Under pathological conditions, the epithelium in any of these situations may become keratinized. Transitional epithelium. This is a multilayered epithelium and is 4 to 6 cells thick. It differs from stratified squamous epithelium in that the cells at the surface are not squamous. The deepest cells are columnar or cuboidal. The middle layers are made up of polyhedral or pear-shaped cells. The cells of the surface layer are large and often shaped like an umbrella. Transitional epithelium is found in the renal pelvis and calyces, the ureter, the urinary cuboidal. The middle layers are made up of polyhedral or pear-shaped cells. The cells of the surface layer are large and often shaped like an umbrella. Transitional epithelium is found in the renal pelvis and calyces, the ureter, the urinary system. With the cells of transitional epithelium are seen to be firmly united to one another by numerous desmosomes. Because of these connections, the cells retain their relative position when the epithelium is stretched or relaxed. At the surface of the epithelium, the plasma membranes are unusual. Embedded in the lipid layer of the membranes there are special glycoproteins. It is believed that these glycoproteins make the membrane impervious and resistant to the toxic effects of substances present in urine, and thus afford protection to adjacent tissues. The cells in the basal layer of transitional epithelium show occasional mitoses, but these are much less frequent than those in stratified squamous epithelium, as there is normally little erosion of the surface. Many cells of the superficial, luminal, layers of the epithelium may contain two nuclei. In some cells the nucleus is single, but contains multiples of the normal number of chromosomes, i.e., it may be polyploid. According to some workers, all cells of transitional epithelium reach the basal lamina through thin processes. If this observation is proved to be correct, transitional epithelium would have to be classified as a pseudostratified epithelium, rather than as a stratified one. Basement membranes of epithelia. We have seen that epithelial cells rest on a thin basement membrane. In multilayered epithelia, the deepest cells lie on this membrane. Basement membranes are formed by thin layers of amorphous material and of reticular fibers. A distinct basement membrane cannot be seen in H&E preparations, but can be well demonstrated using the periodic acid Schiff method. The latter stains the glycoproteins present in the membrane. Under the a basement membrane is seen to have a basal lamina, nearest the epithelial cells, and a reticular lamina or fibroarticular lamina, consisting of reticular tissue and merging into surrounding connective tissue. The basal lamina is divisible into the lamina densa containing fibrils, and the lamina lucida, which appears to be transparent. The lamina lucida lies against the cell membranes of epithelial cells. Membranes similar in structure and composition to basement membranes are also seen in relation to smooth muscle cells, Schwann cells, the glomerular membrane of the kidney, and in membranes covering the cornea and lens of the eye. Several important functions have been ascribed to basement membranes as follows. A. They provide adhesion on one side to epithelial cells, or parenchyma, and on the other side to connective tissue, mainly collagen fibers. B. They act as barriers to the diffusion of molecules. The barrier function varies with location, because of variations in portia. Large proteins are prevented from passing out of blood vessels, but, in the lung, diffusion of gases is allowed. C. Recent work suggests that basement membranes may play a role in cell organoidion, as molecules within the membrane interact with receptors on cell surfaces. Substances present in the membrane may influence morphogenesis of cells to which they are attached. D. The membranes may influence the regeneration of peripheral nerves after injury, and may play a role in re-establishment of neuromuscular junctions. Further comments on epithelia. 1. The shape of epithelial cells is related to the amount of contained cytoplasm and organelles. These are in turn related to metabolic activity. Squamous cells are least active. Columnar cells contain abundant mitochondria and endoplasmic reticulum and are highly active. 2. Laterally, epithelial cells are in contact with other epithelial cells. The contact between adjoining cells is generally an intimate one because of the presence of desmosomes, zonuli adherens, and zonuli occludens. The intimate contact ensures that materials passing through the epithelium have to pass through the cells, rather than between them. 3. We have seen that cilia are present on the free surfaces of some epithelial cells. The surface area of an epithelial cell may be greatly increased by the presence of microvilli, or of basolateral folds. 4. Some epithelial cells contain pigment. Such cells are present in the skin, the retina, and the iris. 5. Epithelia are generally devoid of blood vessels. Their cells obtain nutrition by diffusion from blood vessels in underlying tissues. In contrast, delicate nerve fibers frequently penetrate into the intervals between epithelial cells. 6. Epithelia have considerable capacity for repair after damage. They grow rapidly after injury, to repair the defect. 7. It should be remembered that epithelial cells that look alike, on superficial examination, could have very different functions. For example, cuboidal cells lining follicles of the thyroid gland have very little in common with cuboidal cells covering the surface of the ovary. 8. Epithelia in secretory portions of glands show specializations of structure that depend on the nature of the secretion produced by them. 9. Epithelial cells in which transport of ions is an important function, example renal tubules, are marked by the presence of basolateral folds, and the presence of large numbers of mitochondria. The mitochondria provide ATP, which is the source of energy for ion transport. Tight junctions between the cells prevent passive diffusion of ions. 10. Epithelial cells contain some proteins not present in non-epithelial cells. These include cytokeratin, present in intermediate filaments. Such proteins can be localized using immunohistochemical techniques, see below. Mucous membranes. We have seen that epithelia line many tubular structures within the body. In such structures the epithelium rests, with its basement membrane, on a layer of connective tissue called the lamina propria, or corium. The layer of epithelium along with its lamina propria is referred to as the mucous membrane or mucosa, as its surface is kept moist by secretions of mucous glands. In the intestines the mucous membrane has a third layer formed by a thin stratum of smooth muscle. This smooth muscle is called the MTZSCTZ/AR/S mucosae, equals M muscle of the M mucous membrane. Tumors arising from epithelia. A tumor, or neoplasm, can arise from any tissue if there is uncontrolled growth of cells. Such a tumor may be benign, when it remains localized, or may be malignant. A malignant growth invades surrounding tissues. Cells of the tumor can spread to distant sites, through lymphatics or through the bloodstream, and can start growing there, producing what are called second pharise or metastases. A malignant tumor arising from an epithelium is called a carcinoma. If it arises from a squamous epithelium it is a squamous CE/I carcinoma, and if it arises from glandular epithelium it is called an arlenoma. Quite commonly cells in tumors resemble those of the tissue from which they are derived, and this is useful in pathological diagnosis. However, in metastases of fast-growing tumors the cells may not show the characteristics of the tissue of origin, undifferentiated tumor, and it may be difficult to find out the location of the primary growth. In such cases diagnosis can be aided by localization of proteins that are present only in epithelia. As mentioned above, this can be done by using immunohistochemical techniques.